Hi everyone, I'm Vida. Thanks Kate for the invite, and it's really nice to see a lot of old colleagues here. So I'm a grad student in Joe DeRisi's lab, and our lab focuses on a lot of different types of infectious diseases. So we're a malaria lab, but we also do a lot of novel viral discovery. I think the thing that we all share in common in the lab is we do a lot of genomics, proteomics, kind of large scale big data science. But you know, I like to come to these kind of meetings because I love talking about what it actually means at the end of the day. So how do we actually tackle some of these crazy diseases, and how can we use our data to actually solve some of these really big problems? So I hope that our work actually does filter down and it makes a difference. Okay, so thanks for the introduction. I don't have to talk about the life cycle, but we mainly focus on this intra-erythrocytic developmental cycle. It's 48 hours, and I'm going to focus on kind of these broad stages that are maybe about 10 hours apart. They have the ring forms, the smallest forms in the red blood cell, trophozoites, schizons, and the merozoites are the free extracellular parasites. And there's a lot of work that has been done to actually understand this cycle. So the genome is sequenced, obviously, in falciparum. We've done a lot of kind of genome-wide sequencing to look at different drug-resistant phenotypes to get down to what's the SNP that causes resistance. We also, in 2003, published a paper to look at the actual expression data. So what genes are being transcribed, what's the mRNA abundance at any hour during this 48-hour life cycle. So now you can go onto Plasmodium and ask, when is my favorite gene being expressed? Which is really important if it's a drug target. There's more than just that. So, of course, the central dogma in anything really, in any living organism, you go from the gene, so we have all the information about the genome, to the mRNA, which is transcribed through RNA pol 2, and the mRNA, messenger RNA, gets translated via the ribosome into protein. So we have all the genome information, we have a transcriptome, and then there's some limited proteomics data. Proteomics via mass spec, you get the most highly abundant proteins, so you don't get to see the little ones that you may care about. So there's kind of this gap here. As well, there's been several labs that have seen that there's been discrepancies between protein abundance and mRNA abundance. So when you actually have an mRNA that's being expressed, they actually see no sign of any protein there. So you want to know why is it not being translated. There's also a lot of kind of evidence and little stories that you hear now and then that perturbations such as throwing drugs at a parasite has little effect on mRNA abundance, yet you see huge massive changes in their phenotypes. So what's actually changing? A lot of people are starting to think that something's happening kind of post-transcriptionally through blocking of mRNAs to proteins. And so our lab really wanted to look at this process here, and since proteomics is hard, we wanted to just look at translation. And some notable genes that you'll probably hear a lot about that you already know about, such as BAR2CSA. So this is the ligand that binds to the placenta. And so for pregnancy-related malaria, this is a very, very important gene that's controlled post-transcriptionally. PUF2, this is another protein that works in the sporozoites to liver transition. So this blocks some genes from being translated until they get into the liver. You also have DOSI, which basically also does the same thing, but it's in the transition between gametocytes to oocytes. So these are just a couple of examples that people have worked a really long time to figure out kind of the mechanism that they're post-transcriptionally regulated. But we wanted to go beyond that. So we wanted to look genome-wide. What else can we find? So the great thing about our lab is that we are also right next door to Jonathan Weissman's lab. They're a really great lab that in 2009 developed this method called ribosome profiling. So it's basically a method to isolate and characterize actively translating mRNAs. What that means is that you're actually able to get an idea of what the ribosome is translating, but not due to the protein abundance. This is due to what actually is bound to the ribosome itself, what RNA is bound. And so what we do is we actually grow up these really large flasks. These are 500 mil cultures of highly synchronous parasites, and we take multiple time points. So from rings to, we actually got merozoites more recently. And not to get into too much detail, but you basically make an RNA sequencing background, so you know what the mRNA abundance is. And then you make a sequencing library with anything that's basically a footprint of the ribosome. So what does the ribosome actually sit on? So now you can actually have a measure for active translation. So I'm skipping all the crazy bioinformatics that I've done for this, but what can we learn from this data set? So hopefully, what can other researchers learn from this data set? So just like you go into Plasmodium and you look at what's the expression over a life cycle for your favorite protein, hopefully you can also find, are there any changes on the level of translation for my protein? And there's some really interesting data that we might be getting out with artemisinin also that some of you might be interested in. This might be kind of a really good tool to use. But for example, what can we learn from this? So, this got blocked off a little bit, but this is a sexual stage specific protein. This is PFS16. This is the mRNA, so it's there. It's there during the asexual stages, but the protein's not there. We found a lot of antisense transcription the other way. So is it being blocked, and is that actually repressing translation? Maybe. So there's really cool avenues to look at antisense transcription and how that affects the parasite. We can look at high resolution transcriptional and translational start and stop sites. So you can also see that genes that are on this divergent orientation are highly expressed because they share the same promoter. So those are some really interesting stuff that's going to be available with this data set. There's really cool post-transcriptional regulation. So this is BAR2CSA, like I was talking about earlier. A lot of work has already been published about this. This is already a known gene that's being regulated post-transcriptionally by having the presence of this upstream open reading frame that basically sucks up all the ribosomes, so now you can't translate. So you see evidence of that all over the genome as well. Okay, so what did we find? So this is just wild-type conditions. We didn't throw drugs at it. We're not doing any of that just yet. But what we found going through the life cycle, so this is for you guys that are not familiar with these heat maps, the way you read them are, these are 3,000 genes going down on this axis. So these are all the genes that are expressed during the blood stage cycle. And then on this axis, we have time. So these are from rings, tropes, schizons to merozoites. This is the RNA-seq data. And the color coding is red means that it's highly expressed. Green means that it's very low expression. And typically what you see is a very nice cascade of expression where each gene is only around for that specific time, for that specific purpose, and then it goes away. And so you can also arrange the genes the exact same way looking at your ribosome footprints. And what you see is more or less, for example, let's look at rings. They look more or less the same. Sorry that it may not be that easy to see. But they look the same. Rings, early tropes, late tropes. But when you get to schizons, something really funny happens. And this is something that people have seen in the field or have had hinted to this. That what you see is that these are actually look like the complete opposite. So where you have, for example, high mRNA abundance in schizons for all of these genes, they're really, really low. So they're not actually being translated. Whereas you have some other guys which are pretty low abundance, but they're being highly, highly, highly translated. Translated pretty well. And the merozoites, they go back to normal. So something really weird is happening in schizons. And And so we took a little closer look. And so what are these genes that are downregulated? So if you can plot all the genes, so this is a histogram of all the genes against their translational efficiency, or otherwise, do they have really low translation or do they have really high translation? And what you see is this really interesting bimodal distribution. So there seems to be two populations. There seems to be a population in the cell that is not being translated. It's holding it there. It's there, there's plenty of mRNAs there, but they're not being translated. And then you have this, moderately normal translation pool of mRNAs. And so this is the typical distribution you see in every other life cycle. So what are these genes? Obviously, there's something really important here, because why would you put so much energy into basically making all these RNAs but not translating them? And so when we kind of separate them out to see what these genes are, we actually see that there's a lot of merozoic functioning genes. So GO-term analysis is pretty horrible in Plasmodium, as a lot of you guys know, but what we see are a ton of merozoic genes in the low translation, and then the high translation we see a lot of protein synthesis genes, transcription genes, and so they seem to separate also by function. And so this is kind of ongoing things that we're trying to figure out what these are, but what's our hypothesis? So our hypothesis is that there's translational regulation happening on schizons, where schizons seem to partition a population of mRNAs from low translated to high translated. And so what I think is happening is that the schizons are trying to package up new merozoites. And they need to, kind of stuff in everything in there. So they need to actually, the schizons are in the red blood cell. They've got all the food. They're just trying to package these little parasites together. But they have to make the RNAs for the merozoites to make these new proteins that the merozoites need. But they don't want to express those yet because they're not invading the red blood cell. They're not doing any of those functions. So what they're doing is they're making these transcripts, they're holding them somehow. We don't know what that mechanism is. And they're packaging them, so they're just making tons of proteins just to package. And then once you get into merozoites, all of those transcripts get expressed, get translated, and now you're back into the cycle. So this is really interesting because you got to hold on there's some weird phenomenon happening that's very, very specific at these late stages. So what we'd like to do is to find out what this mechanism of regulation is. Can we perturb this mechanism? Can we find inhibitors of translation? So if this is extremely, this is absolutely essential for the transition into rings, can we inhibit any of those processes? So these are a whole new avenue for drug targets, hopefully. Then can we prevent merozoite invasion via the ribosome? So the nice thing about merozoites are that they're extracellular, so you don't have to go through a red blood cell to hit them with any kind of chemicals or drugs. So if we know more about the basic biology of merozoites, maybe we can actually prevent reinvasion. Okay, so I'm just going to stop there because it's already probably a lot for you guys to think. But I've been working on this project the last few years with Flor and Miguel. Hopefully the paper will be out sometime this year, so it'll be open source and everyone can read it. So thank you so much.
Beautiful. But I'm just curious, how long does the merozoite actually last?
Yeah.
That's the question, because it doesn't even have enough time to make the RNA.
Yeah, so there's some, some people will say that it takes less than 5 seconds for a merozoite to invade. But the thing is that that is, really important because, I'm not exactly saying that you should target just the merozoites, but you should maybe start thinking about schizons all they're doing is getting ready for these merozoites. So maybe there's something mechanistically that you can stop, you can prevent from happening so that any merozoites that actually escape and that have those 5 seconds are already too dead to do anything.
This almost sounds like an unfriendly question. It's not meant to be that way, but I'm just wondering how you're sure that those dramatic differences in, I'll try to say schizophrenia, that it's not due to what could be relatively small differences in the exact stage that we're studying. These are obviously studied by completely different assays.
Yeah, so actually the really nice thing is that you can compare the actual mRNA abundance. And what you see are that all of these genes that are not translated or very low translation, if you look at their whole IDC profile, so their whole 48-hour life cycle, they all exactly start and stop. They look exactly the same for all of those genes. So those are genes that are only being expressed at one certain time. I don't know if that makes sense.
Well, so on the transcriptome level, that would be the same.
They look exactly the same.
Of course, the logical question for your next year after you write that paper is going to go to the protein level.
Yeah, exactly. And so I actually have, sorry, I didn't add this to the talk, but I've been doing stage-specific in vitro translation where you can take extracts of parasites, and now you can play with the ribosome. You can actually test whether something, if I throw in another protein factor, how does that affect translation? Now, do I get translation of those low expressed genes? Do I get, de-repression, any of those kind of games, complementation games, drug games, with not a whole parasite, but with looking specifically at translation?
I just have a general question. How often do you see these bicistronic mRNAs? Because I know there are some common ones that are bicistronic in these falciparum proteins. Do you ever see those two proteins expressed from one RNA?
Yeah, so the RNA-seq that we do, they're really short, so we actually are only doing 30-mer sequencing, so we don't actually see much of that. But some interesting things that we do see is that you get a lot of just transcription all over the place. So you get transcription from backfiring from promoters, and that actually causes a lot of these antisense transcripts. Transcription kind of is not very well regulated, it seems, in malaria. So they just transcribe everything, almost.
You've probably worked all of this out, so I'm sure you have a good answer, but thinking about these antisense transcripts, how do you know when you grind up a parasite that piece of RNA was supposed to be stuck to that ribosome? Because you're pulling down the ribosome, right? And it's
So that's really interesting. So what you actually can get is, so we do the nuclease digestion, and so you have protection of these transcripts by the ribosome, and that are very specific to a certain size. So the size of the ribosome is about 30 nucleotides. And so you can actually do a read length distribution of all your footprints versus everything else. So you can get a distribution of your rRNA, of your tRNA, and those are completely different than everything we see that's in the coding region. And antisense transcription is very much the same kind of distribution as the coding region. So you can get an idea of whether they're actually being protected by the ribosome or whether they're actually just stuck somewhere outside that may just come down with your whole prep. So yeah, you can actually look at your specific gene and see what the read-link distribution is.
Have you analyzed the group of these transcripts for any common features that might suggest a common transcription factor or?
Yeah, so for transcription factors, so we actually, the way that we do our library prep is a little bit different. So we're not getting all the way to the promoter. I think we're getting really close, but we have another grad student in the lab who's trying to get to the very end. And he's starting to look at this data set and analyze changes in the length of 5' and 3' UTRs and how that affects maybe possibly the exposure of a new binding site for RNA binding protein or something like that. It's still in the works, but once we get this paper out, then we'll start looking a lot harder.
One more question. What do you think it is? What do you think is keeping them from being translated inside those chizons? What's your just intuition?
So my intuition is that there's a mark that's probably cis-acting elements on the RNA. That seems to be kind of an overall theme in post-transcriptional regulation in malaria. So probably something on the 3' UTR where I have to start looking for motifs because it's a really nice set of genes. It's like a 500 or so genes, and they're all regulated in a very similar way. So there's got to be a motif in there, or somehow marking those transcripts. One thing that we are going to do is we're going to start doing some RNA fish to see where are they localized within the cell. So are they actually being sequestered in a P-body like the puff proteins do, or are they stuck to another organelle or something like that? Yeah.
They're really these are late Merozoic proteins. A number of them are under immunologic pressure, which is unusual for the rest of the genome. So this you may be hinting at a mechanism of some method of plasticity to allow changes post-translational or whatever that allows immunogenic diversity of the parasite.
Yeah.
Right.
Okay. All right. Thank you. Thank you so much.